In this lecture, we will talking on how these elements, which are other than the TN elements. In TN elements, we have the different patterns of the resident genes and multiple insertion sequences, composite sequences, but here insertion sequences are the simple sequences. So first we talk about the insertion sequences, then we talk about the composite transposon. Now, the simplest transposable elements are insertion sequences we call IS element, which is consist of a short DNA of almost 1 kb to 1.5 kb base pair sequences. A single gene encodes the enzyme transposase. So this insertion sequences carry the one gene which is coding the transposase, and terminal inverted repeats like 15 to 25 base pairs at both ends. Now, each IS elements is named with the prefix IS, like insertion sequences, followed by the number. Say for example, IS2, IS10, IS125 and so on. Now, the transposase gene which is present in the IS elements recognizes the inverted repeats, right? Inverted repeats means like, say for example, this is the insertion sequences. This is the inverted repeat. Now, this inverted repeat is basically responsible for inversion of this gene, or if it is direct repeat like this, it causes them to delete or to insert. So this is the function of IS elements, depending upon what kind of a structure of the IS element has, whether it is a inverted repeat flanked by inverted repeat or the direct repeat. Now, each IS element is named, okay, the transposase gene which is present in is recognize the inverted repeats and then catalyze it to transposition at the movement of elements. Now, IS elements have been observed in many bacteria, in some archaea also. They can cause mutations by inserting into genes or regulatory regions. Now, just have a look at the diagram. The insertion sequences, this is the insertion sequences, and you have a transposase gene here. This is the inverted repeats, two inverted repeats, this is the two direct repeats, this is the whole chromosomes. Now, depending upon the inverted and direct repeats, you can fate of the IS element, whether it has to switch to other chromosome or not, or they form another molecules to recombine in other chromosomes or not. So this is decided by these repeats. And this is composite transposon. This is the replicative transposon. We'll talk about the composite transposon and what is the replicative. So composite transposons are the transposons. Basically, composite transposons contain gene in addition to those required for transposition, such as antibiotic resistance or toxin genes. They typically have two structures. IS flanked composite transposons and simple composite transposon. IS means insertion sequence flanked composite transposon, in which you have two insertion sequences like in flanking regions, and then we have a transposon. For example, if this is the transposon, then these are the two insertion sequences like this, and then this insertion sequences could have its own inverted repeats, and this insertion sequences could have its own inverted repeat. Depending upon these insertion sequences, the transposons carry the gene is being transferred, or is being inverted, or is being switched to other part of the chromosome or to other organisms like in other bacterial species. So the central regions contain the additional gene flanked on both the sides of IS elements that are identified through the repeats are very similar to the sequences which are already identical to these two inverted and IS elements. The IS elements provide the machinery for transpositions because it has a transposase gene, right? And they help in transposition to the transposon. Now, the simple composite transposons are those which are bounded by the short inverted repeats, right, with coding regions containing both transposition genes and additional functional genes. So, in which no IS element is present, but the simple inverted repeat are present, because ultimately it is not the IS element but the inverted repeat are required. But since IS element when present, they have transposition encoding gene, that is transposase encoding gene, which help in the transposition. Therefore, that helps, this work is being done by some other protein along with the inverted repeat in the simple transposition. The formation of composite transposons likely of two IS elements associated with the central segment of gene which contain certain resistant genes, and this could happen if an IS elements replicate and inserts only short distance away from the original location. So this is how it forms. So if you look this like example, this is TN3. This is the length of TN3, almost 5 kb. Then we have a terminal repeat which is 38 base pairs, and this is genetic markers like ampicillin resistant marker. Now in TN501 we have almost 8 kb. Again we have a mercury resistance. Then we have a TN1681, 2 kb. We have a heat stable enterotoxins, and TN2901 almost 11 kb, which has IS1, and arginine biosynthesis. So these are the different category, different type of transposon which are characterized by having different type of genes in it, right, and different type of resistant markers are gene, and they are having different size of the transposon depending upon the IS element presence or absence of IS elements.